Good morning, ladies and gentlemen. My name is Pavla Bauerova, and I work at the Laboratory of Scanning Electron Microscopy here at UCEEP. My job today is to tell us something about what we do in our laboratory. I would like to introduce some projects that we are currently working on. So now let's start. I've got a feeling that scanning electron microscopy is a bit aside of all the other things that are carried out here at UCEEP. So I would like to introduce the device itself at the very beginning. Scanning electron microscopes are actually very powerful devices which can see much more than not just the human eyes, but more than light microscopes. That's why these devices are almost in every lab, in every field of research. Whose goal is to see inside the inner structure. The big difference between a scanning electron microscope and a light microscope is that instead of light, we use an electron beam for magnification, which you can see in this picture. There is also a set of magnifying lenses, but the difference is that we use a highly accelerated electron beam. And also the difference between an electron microscope and a classical light microscope is that the radiation doesn't go through the sample, but it scans the surface of the sample, which you can see here. It scans across a certain area, point by point, line by line. That's why it's called scanning electron microscopy. A signal is generated. The signal is connected to the detector, and from the detector, the signal is processed to a computer. And the result is a black and white image of the structure. Now, this device can provide much detailed resolution. You can see the comparison here. In fact, the microscope can see, as far as I calculate, right, 200 times better than a human eye. This is the microscope that we have here at UCEEP, and this is the microscope that we use for our experiments. Now, how it works. When electrons hit the sample, a wide range of interactions take place. For us, the most important are the three of them: the generation of the so-called secondary electrons. The images, secondary electron images give us an information about the microstructure, about the topography of the sample, what the sample looks like. Then backscattered electrons, this signal can give us an information about the material composition, very general, whether if there are two materials in the sample, this backscattered image can distinguish which material is lighter and which is heavier. But the microscope can also provide a complete chemical composition analysis. It uses characteristic X-rays that are also formed during the collision of the primary electron beam and the sample. This microscope that we have here at UCEEP is unfortunately not designed for the observation of inconductive samples, and building materials are inconductive samples. So the preparation of the samples is a bit more challenging than it normally could be. We have to coat them with a conductive layer, and we also do cross sections. We embed the samples of building materials into polymer resins, grind them and polish them before we put them in the microscope. Now to the projects that I would like to introduce. The first one is the study of the properties of thermally loaded gypsum. Gypsum is a very common material, building material used for mortars. Unfortunately, Its disadvantage is that its mechanical properties are changed when the material is exposed to temperature. And scanning electron microscopy can provide an information why it happens. You can see the graph of the changes of the strength of gypsum pastes. You can see that when the gypsum material is thermally treated to certain temperatures from 50 degrees to 1000 degrees, now between 50 and 100 degrees, there is a significant decrease of the quality of the mechanical properties, and it actually continues until up to 900 degrees. And when you look at the images above, you can see that the explanation, one of the explanations lies in the structure. You can clearly see in these images that the structure of gypsum is changing. Yeah, these are the secondary images which give us an information about the topography, what the sample looks like. So this was one of the examples. Another one is a project carried out by Zdeněk Prošek, by a colleague of mine, who investigates the self-healing properties of concrete. He wants to, or he and his colleagues want to investigate the effect of bacteria, of calcite precipitating bacteria on the quality of the cement pastes. Again, I will show you the pictures, and you can see several pictures. This one is a picture which is made where no bacteria and no bacteria supporting medium was used, just simple water during the preparation of the cement paste. And these images, they are the pastes based either on the medium or medium with bacteria. And you can see, if you compare these two images, especially these two details, there is a significant difference between the size of the crystal formed. You know, I should have told you at the very beginning that this is a cement paste, and there were experimental grooves made in the paste, and they investigated how the groove is filled, either spontaneously or with the help of the bacteria. So here you can see the grooves, the cuts in the material, and you can see that in the images where the bacteria were applied, the grooves healed faster, better. They are not so clearly seen, and moreover, the calcite which is produced during this process is it has much smaller grains compared to the spontaneously formed calcite. This is important because in the literature, in the previous literature, it was previously reported that there's no way how to distinguish the biogenic calcite formed by the bacteria and the spontaneous calcite. This was because these claims were because the scientists did not use electron microscopy in their research, but other methods. Another example is the utilization of waste marble sludge, which is considered to be added as an admixture. It's like a sort of environmental project. And they evaluated whether the addition of the waste marble sludge has a positive or negative impact on the hydration of cement. And in these images, the very shiny spots are clinker residues, unreacted cement. When the cement is hydrated, when the cement is hydrated, it means that the clinker is disappearing. When you compare this image where no waste marble sludge was added, and the image of 15% addition of waste marble sludge, you can see that there are, well, if I say it in a very primitive way, there are less shiny spots in the images, which means a better hydration reaction of the clinker. And so it can be concluded that waste marble sludge improves the hydration of the clinker, and it appears to be a promising material. It could be processed this way. And last but not least, my favorite project is the analysis of historic mortars. We focus on the investigation of the fixing mortars in historic mosaics. And here you can see some examples of the mosaics that we currently work on. We took samples of them, and we identified several different materials, including lime, Cocho pesto. It's an interesting picture. You can see this is the map of the elemental composition, and here you can see an inhomogeneous particle, which is a brick. I don't know if it's clearly visible, but the brick itself is also inhomogeneous. You can see brown quartz grains and the violet aluminum silicate. And the green is the calcite matrix. And the lower images are the mortars of this mosaic, and this mosaic is based on Portland cement. You can see a clinker characteristic for Portland cement. So, to conclude, now I cannot skip to, yeah. To conclude, scanning electron microscopy is a very powerful tool for the analysis of microstructures and elemental composition of building materials. I think we reached quite interesting results. And that's it. So thank you very much for your attention. And if you got interested in what we are doing, you are welcome in our laboratory for a kind of excursion. Thank you very much.